Before diving into the figure panels, this slides outlines the experimental design used to visualize mechanical tension and cell polarity during dorsal appendage morphogenesis in the Drosophila egg chamber. The two key fluorescent markers were used in this experiment. First one is SQHGFP, a fusion of the regulatory light chain of non-muscle meson. It shows green here, and it's tagged with GFP, serve as a readout for cortical tension. The second one is called bazooka. It's a polarity protein analogous to PAR3 was detected using immunofluorescence. To capture the spatial and temporal dynamics of these proteins, confocal microscopy was employed to image both apical and basal surface of the epithelial primordium across developmental stages. These sacs were reconstructed into top-down or oblique views to reveal the distribution of myosin and bazooka during morphogenesis. The 10 points shown in panels C to G and their corresponding basal views C prime to G prime were selected to represent key morphological transitions, including sheet bending, floor cell intercalation, and seam formation. And in the slides one here, picture A to A prime. It describes that pre-patent tension predicts morphogenetic zones. In this first panel, we observe the dorsal appendage prim, primordium at an early development stage. The green signal represents SQHGFP, a marker of apical cortical tension, while magenta shows bazooka, a polarity marker. Notably, we see two clear boundaries enriched with meson, the floor roof and the floor midline interfaces. At these same locations, bazooka expression is reduced, creating a complementary pattern. This suggests that mechanical tension is not uniformly distributed, but instead spatically patterned early in development. These pre-patterned line tensions likely define zones of future morphogenetic activity, such as um, epithelial bending and cell intercalation. Uh, Figure 2, B to B prime. Uh, it shows that localized line tension identifies mechanical boundaries. Here we zoom in, examine the apical surfaces of individual epithelial cells. The SQH-GFP signal forms a sharp, continuous line along the floor midline boundary, while bazooka is again reduced in this zone. This close-up demonstrates that the mechanical tension mediated by meson is tightly localized at the specific junctions, and that these sites of tension correspond to bazooka exclusion. The possession of this pattern at the single cell level supports the idea that specific edges within the tissue are mechanically distinct, providing directions cues for how the tissue will bind and rearrange. Slide 3 is talk about C to picture D. Um, it describes that dynamic reorientation of tension drives the morphogenesis. This panel series tracks the apical surface of the primordium from early to late stage. In C picture, SQHGFP is concentrated along the roof boundary, matching early observations. As we move through D to picture F, this rearrangement shifts toward the midline. Um, by G, meson accumulates along the seam where floor cells meet. This dynamic redistribution suggests that apical tension is not static. Instead, it is redirected as the morphogenesis process progresses. The shifting location of tension likely guides the sequential steps of bending, cell intercalation, and sealing. Slide 4, picture C prime to G prime is talk about tension refocuses toward apex for film formation. This companion set of panels provides upside up-down basal views, like corresponding to C to G picture stages, allowing visualization of tension dynamics from the underside of the tissue. Initially, SQHGFP enrichment is seen along the basal regions near the roof boundary. As development progresses, the tension focus shifts ventrally and anteriorly toward the midline. By panel G, the strongest signal appears near the anterior apex, aligning with that where the two floor cell sheets will meet the seal. This image reinforces the observation that contractile forces are reoriented and focused toward the apex throughout morphogenesis. Slide 5 is talk about boundary alignments and Mason accumulation stabilized tuber closure. In this final panel, we observe panel E double prime, F double prime, and G double prime in sequence. These images focus on the basal surface of the primordium. E double prime and F double prime show the progressive alignment of cell boundaries as the two arms of the floor cell domain move toward convergence. This structural organization anticipates the final step, showing G double prime, where SQHGFP becomes highly enrichment at the apex. This terminal accumulation marks the peak of the mechanical stress and indicates the site of tube sealing. Together, this panel highlights the coordinated rearrangement and tension accumulation that mechanically stabilize the tubular morphology. And in the entire figure 4 provides a very detailed spatial and temporal map of cortical tension and the polarity protein distribution during Drosophila dorsal appendage formation. The experiments demonstrate that morphogenesis is driven not by uniform tension but by dynamic localized enrichment of myosin along specific cell junctions. These tension patterns are established early, evolving through morphogenetic progression, and ultimately uh, culminated in the mechanical closure of the tube at the apex. Together, this data support a model where the spatial and temporal control of line tension is sufficient to guide the transformation of a flat epithelial, uh, ep- epithelial sheet into a complex 3D tubular structure.